Hi everyone. Hi everyone, welcome back to my channel. This is Prashanti, your biology teacher. So in today's class we are going to see from the CBSE board, okay, CBSE board class 10th biology. So from class 10th biology the lesson is about heredity and evolution, okay, heredity and evolution. So fastly I am going to explain what is heredity and what is inheritance and what are genes. Okay, first, what is heredity? So heredity is nothing but passing the characters from parents to progeny. Parents to progeny is nothing but from parents to offsprings. The young ones are receiving some characters, not all the characters. Why? Because here if you see in the sexual reproduction, especially, so the offsprings produced by the parents are not similar. Exactly, they are not similar, though they are having the same parents, but the characters are different. They will show variations, okay. But at the same time, here that progeny, the offspring generation, the next generation is receiving some characters from the parent, and this whole process is known as heredity, right? This process is known as heredity. So what are the characters we'll get maximum from the parents? So that may be height of the body, and at the same time color of the body, hair texture, and at the same time hair shape. Maybe some offsprings or some progeny may have the straight hair, some may have the curly hair, some may have the wavy hair. Wavy is nothing but in between the straight and curvy. So this type of characters we'll get. And sometimes ear shape, sometimes the ear will be closely attached to the skin, sometimes it may be free, okay. So this type of characters we'll get. And also one more thing, color also will get from the parents. So this type of characters are going to be passed from the parents to progeny, and the whole process is known as heredity. That's it. And what are the factors are responsible to pass these characters from one generation to another generation? Those factors are known as genes, right? The functional units of heredity, the functional units of genetics are genes, okay. So those genes are nothing but the structures which is responsible to carry the characters or traits from the parent generations to the progeny. These genes are nothing but they present in the DNA, okay. So and then today we are going to see one topic from the heredity and evolution. That topic is about dihybrid cross, okay, dihybrid cross. So for more biology classes of mine, you can subscribe my channel, and for updates, for continuous updates from my, so you can able to click the bell icon. Please click the bell icon and subscribe the channel so that you will get more interesting biology classes from me, okay. We'll start the session. Dihybrid cross. So dihybrid, di means two. So here in the name only we have the answer. What is the answer? Dihybrid is nothing but two characters are considering. Here we are taking two characters from parents, okay. So two characters of two organisms are going to be fertilized, are going to be pollinated. If it is in the plant condition, we will pollinate the plants, and in the animal condition, fertilization is going to take place. So here this we are taking the plants, pea plants, so that's why we are pollinating. So how the pollination process is going to take place? So in the dihybrid cross, every time in the monohybrid or dihybrid, we have to think about one thing. First we are doing, so here we are not doing, the Mendel did the experiments. So Mendel how he conducted the experiments? So Gregor John Mendel, he is the father of genetics. So So without any scientific equipment, so in that time only in the 18th, so he had conducted many wonderful experiments to give knowledge about the genetics and genes and characters, everything. So Gregor Jan Mendel conducted the experiments regarding the pea plants. So here he has selected two pure breeds. You have to know about what are pure breeds. Pure breeds are nothing but after fertilizing, after pollinating many times also, if the plant is getting the same character. For example, if yellow flowered plant is pollinating many times, after 15 generations or 11 generations or more than that, if the next generation plant is getting the same yellow color, so that type of plants are known as pure plant. Okay, pure breeds will say. So that type of pure breeds he had selected for this experiment. What are the two characters he has selected here? The two character he selected, one is C, round and yellow. Capital R indicates round character. Capital Y indicates yellow character. Okay, so capitals always indicates the dominant traits. Even in the introduction also I have told that in the heredity, so dominant traits always is going to be denoted with the capital letters, and recessive trait means, so they are not going to be expressed in capitals, they expressed in small letters. Okay, so dominant is nothing but, one more time, listen carefully here. Dominant traits are nothing but, what are dominant traits? Here you will get 1 mark or 2 marks answers from this dominant traits and recessive traits also. See, listen. So dominant traits is nothing but the characters we'll get in the F1 generation. So the character after cross pollinating two breeds, see here small diagram. So first one, this is one character, so round, and here and one more character is wrinkled. Okay, so round character and wrinkled character. So we are going to be cross pollinating. So we are doing cross pollination here. After cross pollination, what we'll get? We'll get a hybrid. So what is the hybrid here? Sorry, write with smaller and smaller. So in the F1 generation, automatically in the presence of one character, see here capital R, which is going to be expressed in the F1 generation, that type of characters are known as phenotypic characters. Phenotypic characters means maximum they are going to express themselves. That type of characters are known as phenotypic characters. So here the phenotype character is round. Okay, so with the presence of one capital R also, that complete breed or offspring will get the same character. So that is what F1 generation means. This is F1 generation. F1 generation means what? Here, when the cross pollination takes place between two pure breeds, the next progeny, the hybrid, is going to get one character among themselves. So that character will be the dominant trait, and the character which is not expressed, and that will be the recessive trait. So here you have this 1 mark and 2 marks answer also. Dominant traits and recessive traits. What is dominant? Which is expressed in F1 generation. What is recessive? Which is not expressed in F1 generation. Okay, so how can we determine the dominant character and recessive? In the presence of the capital letter or capital factor, so that is going to be capital R or capital Y or capital T. In the monohybrid cross, we have taken length, height of the plant. So in that height of the plant, two characters, one is tall and dwarf. Okay, tall and dwarf. tall is going to be indicated with a capital T, and dwarf is indicated with the small t. Okay, so based on that information, we are going to see this Punnett square. See, every time the pollination boards we can able to check in the Punnett squares. So okay, this square is known as Punnett square. Write this point, Punnett square. So in this Punnett square, we have taken 4 characters. Okay, 4 characters. So in the presence of capitals, see, round and yellow, R, capital R and capital Y. So anytime in the breed, if there is one capital R or one capital Y, that is showing that characters. Okay, that is showing the character. See here, the chances, probabilities we are seeing in between the 2 characters. The dihybrid is nothing but, so 2 characters are going to be considering. One character is about round yellow, another character is about wrinkled green. Okay, wrinkled green. So when the self pollination in the F2 generation, we are doing the self pollination. So in the F1 cross pollination, but later we are doing the self pollination. In the self pollination, so these are the probabilities. So first one, capital RY, same characters. See, one side, anytime when we are writing in the Punnett square, same characters we are going to write in the horizontally and vertically. See, and the symbols indicates, see the symbols. So here this symbol indicates female and this symbol indicates male. Okay, so the cross pollination is going to done between the pollination done between the male and female. So here the pollinations same with the characters, capital RY, and here also capital RY, capital R and one small y, capital R and one small y, and smaller r, small y, and smaller capital Y. And the possibilities, so probabilities will be, so see here what I said, in the presence of capital R and capital Y, that is exhibiting the same character. That's why, see, all yellows, so all yellows and rounds, so yellow and rounds indicates in the presence of one capital R and capital Y. So ekkadaina one capital R one capital Y undante, it is exhibiting round yellow character. అదొక్కటే మీరు ప్రాక్టీస్ చేసేటప్పుడు గుర్తుపెట్టుకోండి. So next possibilities, see, so these are the probabilities for round yellow. And then see here, 2 capital R's and 2 small y's, 2 capital R's and 2 small y's. 2 small y's indicates green color. So 2 small y's indicates green color. So automatically, so here the green color is dominant and round is dominant. Automatically the P will be round and green. Round and green. How many are there? 1, 2, 3. 3 round and green. And next one, see, one more combination, 2 small r's and 1 capital Y and 1 small y. So in the presence of one capital Y, it is expressing its character, and it will be the wrinkled yellow. Wrinkled yellow, it is not round, wrinkled. Okay, wrinkled yellow we can see. So how many are there? 3 are there. Okay, and one more combination, see, presence of 2 small r's, 2 small y's. 2 small r's and 2 small y's means here both are expressing its character. So one small r indicates wrinkled, small y indicates green, wrinkled green. So what are the characters we are getting here? Mainly 4. First one, round yellow. How many round yellows are there? 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 round yellows. So the ratio will be 9 round yellows. 9 round yellows. And then how many round greens? 1, 2, 3. 1, 2, 3. So 3 round green. Again, next, how many yellows are there? Wrinkled yellows, 1, 2, 3. Wrinkled yellow, 3. How many wrinkled green? 1. Wrinkled green is 1. So what is the ratio here? 9:3:3:3:1. This ratio is known as dihybrid ratio. And here which law is going to be followed here? Law of independent assortment. Law of independent assortment is nothing but here independent assortment. One more time I am explaining law of independent assortment. That is here in this pair of characters. So in this pair of characters, the factors are expressing or assorting independently their characters to the offsprings. So this law is known as independent assortment. Law of independent assortment is nothing but in the among the pair of alleles. So these factors also known as alleles. Okay, so this one pair of alleles, it can able to assort its character or trait independently to the offsprings. So this law is known as law of independent assortment. Okay, that's all for today's class. So that is about dihybrid cross. So I'll be back with the another topic from the heredity and evolution. So keep watching and at the same time subscribe and like this session. Okay, thanks for watching this.